Hello, this is Professor Torsen. This is the recorded lecture for Topic 5, the nervous system. The nervous system has three overlapping functions. The nervous system is in charge of gathering information from the sensory receptors and processing that information. It is also responsible for designing and producing the response to the sensory input. So for example, in this simple figure here, part of our nervous system is gathering information from the environment. So we see that there is a glass of water, and that information is sent to the nervous system. Here we're seeing an image of the brain. The brain integrates that sensory information with ongoing information. For example, are we currently thirsty? Maybe we just ate something very salty, and designs a motor response, which here is picking up the glass of water so that we can drink it. Communication within the nervous system is very quick. Cells within the nervous system utilize electrical and chemical signals, and these are very quick. Before we get too far into how the nervous system functions, I want to discuss the divisions of the nervous system. The divisions of the nervous system include the central nervous system, or CNS, and the peripheral nervous system, abbreviated PNS. So the central nervous system, or CNS, includes the brain and spinal cord. The peripheral nervous system, or PNS, is basically everything outside of the brain and spinal cord. So the peripheral nervous system includes spinal nerves and cranial nerves. And we are going to look over the next couple of slides a little bit more closely at the sensory and motor divisions of the PNS. So again, the peripheral nervous system can be divided into the sensory division and the motor division. The sensory division, also called the afferent division, consists of sensory fibers. So we have somatic sensory fibers that convey impulses from our skin, muscles, and joints. We also have visceral sensory fibers that convey information from visceral organs. All of this information is being carried from the peripheral to the central nervous system. The motor division, also called the efferent division, transmits information from the central nervous system to the effector organs, typically muscles or glands. The motor division is called the efferent division because efferent begins with E, and this information exits the central nervous system. So E for efferent, E for exit. The efferent division can be further subdivided into the somatic nervous system and the autonomic nervous system. The somatic nervous system is the voluntary nervous system. So that is our conscious control of skeletal muscles. The autonomic nervous system is involuntary. So we don't control the autonomic nervous system. Autonomic sounds like the word automatic. So autonomic nervous system automatically happens. We don't consciously control it. The autonomic nervous system includes the sympathetic and parasympathetic nervous systems. So in this figure, we can see the breakdown of the classifications of the nervous system. So starting at the top here in red, we see the central nervous system includes the brain and spinal cord. The peripheral nervous system, which is essentially everything but the brain and spinal cord, can be divided into the sensory division and the motor division. And we can see the motor division is further subdivided into the somatic nervous system, which is under conscious control, and the autonomic nervous system, which again is not being consciously controlled. Now that we have the various divisions of the nervous system, let's look at the different cell types within the nervous system. The two main cell types of the nervous system are neurons or nerve cells and neuroglia or glial cells. So neurons are excitable cells that are able to transmit electrical signals. Neuroglia or glial cells are supportive cells. So glial cells aren't transmitting specific information the way neurons are. They are working to support the neurons. So on this slide, we see 6 different glial cells. 4 of them are in the central nervous system and 2 of them are in the peripheral nervous system. Let's begin by looking at neurons. Neurons are special cells of the nervous system that are able to conduct impulses, electrical and or chemical signals that relay information. We've discussed the ability of various tissues to regenerate or repair themselves. Neurons have a very low regenerative capacity. For the most part, neurons are not able to divide. They are amitotic, which means that any injury to the area, like in the case of traumatic brain injury, or in the case of a neurodegenerative disease, once those neurons are lost, there's very little our body can do. Our neurons are designed to last the person's lifetime, and so anything that damages neurons is really hard to treat. Neurons have a high metabolic rate. They require a lot of oxygen and energy, glucose. In terms of neuronal cell structure, you can see a typical cell here on slide 7. Here in purple, we see the nucleus. And this yellow portion of the cell is the soma, or cell body. The cell body contains the nucleus of the cell. Surrounding the soma are these yellow thin branches. These branches or processes are called dendrites. Dendrites are the receptive region of the neuron. Dendrites will receive information from other cells. Towards the bottom, you see the axon, which is the other main projection coming off of the cell body or soma. The axon is the conducting or output region of the neuron. The axon is the conducting or output region of the neuron. We see in this figure here the cell body or soma with all of the dendrites that are receiving information. And here towards the bottom is the axon. The axon is actually this yellow part here. You can see the end of it here on the right-hand side. The blue part is actually myelin, and myelin is formed from glial cells. So let's look a little bit closer at myelin. If we look at this cross section of an axon here, we see this center region labeled axon. So this is part of the neuron. This darker ring surrounding the axon is myelin. Myelin is actually part of glial cells. Myelin helps to protect and insulate the axon. Insulating the axon means that the speed of information, or the speed at which information can travel down the axon, is faster. So non-myelinated fibers conduct information much slower than myelinated axons. This is important when you consider some neurons, axons are a meter or even longer. So they are traveling quite a long distance. And if we didn't have myelin insulating the axon, the signal would take a very long time to reach the terminal regions. White matter and gray matter are terms used to describe part of the central nervous system. White matter appears white due to the large amount of myelin present. Gray matter appears gray because it is mostly those neuronal cell bodies, those somas, as well as non-myelinated fibers. So again, myelin helps insulate axons, and myelin is formed by glial cells. In the peripheral nervous system, myelin is formed by Schwann cells. In the central nervous system, myelin is formed by oligodendrocytes. Both Schwann cells and oligodendrocytes are glial cells. Here you can see figures of an axon in yellow, and in blue you see the wrapping of myelin around the axon. Neurons can be classified based on their structure, and their structure is reflective of their functions, which is something we talked about at the beginning of the semester. Cells and tissues have a specific structure because they need that structure in order to perform their function. So there are bipolar neurons. You can see that in this figure here. Bipolar, bi meaning two, have two projections coming off of the cell body. Unipolar, uni meaning one, unipolar neurons have one projection coming off of the cell body. So here is the soma or cell body, and here is the one projection. Now this projection is splitting, but they both come from the same part of the soma. So this would be a unipolar neuron. We have many multipolar neurons. Multipolar neurons have several projections coming off of the cell body. So here you see 6 different projections coming off of the cell body. Again, the cell type of the neuron is dependent on the function that it is trying to carry out. Neurons have a membrane potential. Membrane potential is the voltage, essentially the different charge inside versus outside the neuron. So we're really interested in the ions and the concentration of ions inside versus outside a cell. So in this figure here, you see inside the neuron, we actually have a fairly high concentration of potassium, K+, There is a little bit of sodium, or Na+, and there are plenty of proteins that are negatively charged, which is represented by this letter A. Outside the cell, or extracellularly, there is a predominance of chlorine, which is negatively charged, and sodium, which is positively charged. There is a little bit of potassium outside the cell as well. So it's these differences in concentrations of these ions inside versus outside the cell that is responsible for the membrane potential of an. As I mentioned on the previous slide, the membrane potential of a neuron is largely determined by the ions that are present inside versus outside the cell. So what we're talking about here is the electrochemical gradient. Not only the concentration of various ions, but we also need to take into account the electrical charge, because remember, these are ions that have charge. A quick primer on electricity is shown on slide 14. So there is Ohm's law, which shows you the relationship between voltage, current, and resistance. Now, I'm not going to ask you to perform any calculations using Ohm's law, but this cute little figure here, I think, illustrates Ohm's law and the relationship of these variables. So again, what we often measure and what we're going to be looking at in the rest of these slides is membrane potential, which is the voltage of the cell. So this yellow guy here. Current is the flow of electrical charge, so the movement of ions. And that is being represented by this green guy here, labeled amp. Resistance is this red guy here with the ohm hat. Resistance can hinder the flow of ions. My example for this is a garden hose. So in your yard, you might have a hose for water, and the flow of water is dependent on the diameter of the hose. So if I pinch the hose, if I bend the hose and I pinch it down, the water starts flowing much more slowly out of my hose. And then the pressure starts to build up. And then if I release this pressure or this resistance by releasing the bend in the hose, then the water can flow freely. Ions are unable to diffuse directly through a cell membrane. Instead, for an ion to cross the cell membrane to either enter or leave the cell, they need some way to get across. So we have large transmembrane proteins called ion channels that can open and close to allow ions to flow across the cell membrane. These ion channels are tightly regulated. Some of them are chemically gated, also called ligand gated. Chemically gated channels only open when a certain chemical binds to that gate or channel. So for example, a neurotransmitter might bind to an ion channel and cause that ion channel to open and allow flow or movement of ions. You can see that in this figure on slide 15. Here is the ion channel. It is closed, and neither sodium nor potassium can move across this cell membrane. However, after a neurotransmitter has bound to the ion channel, and again, this is a chemically gated ion channel, the neurotransmitter binding to that ion channel causes it to open, and now we can see ions are able to flow or move across the cell membrane. Some ion channels are voltage gated. So voltage gated ion channels open or close in response to changes in membrane potential. And remember, membrane potential is the voltage of the neuron. We do also see mechanically gated channels, and mechanically gated ion channels open or close in response to physical changes to the receptor. And we will see this when we look at the sensory system more closely in topic 6.